Hey guys. So we're gonna go over, sorry, let me hide this. We're gonna go over just a few biotechnology techniques. So like there's a lot, but there's only a few, oh, excuse me, only a few we really need to know. You don't need to know like the specifics for every one on like how exactly they function, but you should know what each one does. And then what they'll do a lot of times is they'll provide you an FRQ, and then like within that FRQ, they will have like one part asking with like a genetic engineering technique and stuff like that. So the first one I wanted to go over was restriction enzymes. So you should just simply know that restriction enzymes are enzymes that cut DNA at really specific sites. So if I wanted to take a piece of DNA and if I wanted to cut it so that I could remove a particular gene from it, then I would have to make sure I cut at the beginning of that gene and I cut at the end of that gene, and then I can remove that gene from that DNA sequence. And then you can put that gene into something else and create what's called a vector. So we use what are called restriction enzymes, and there's many different restriction enzymes. And what they do is they are proteins. And what these proteins do is they recognize, it says right here, palindromic DNA sequences. So they look for a DNA sequence that's a palindrome when you read it frontwards and backwards from the like 5 to 3 prime, and then down here 5 to 3 prime. So when we say palindromic, I don't mean that when I read it this way on the 5 to 3 and then read it this way from the 3 to 5, it's the same. What we're saying is that on one strand, it'll read GAATTC from 5 to 3. And then on the other strand from 5 to 3, it reads GAATTC. So you have palindromes on the alternate strands of DNA. So what happens is, in the example they're using here is called EcoR1. So EcoR1 is a really specific restriction enzyme that we use a lot to cut DNA, and it cuts it always at this sequence. So it searches for GAATTC, and it always cuts between the G and the A, so the guanine and the adenine right here. So if you expose DNA to this restriction enzyme, it'll just zip along DNA like so. And when it finds this palindrome, it binds to the palindrome, and then it will snip or cut the covalent bonds right here between those two nitrogenous bases. And you can end up with two types of ends. So if it cuts it straight through, so if it was cutting between the A and the T, it would cut like straight through the DNA. So you would have what we call blunt ends. But if it cuts like this, where you get this overhang, like so, and you can kind of put the DNA back, this is what we call sticky ends. And so we can cut DNA like this and produce sticky ends. And then I can take this piece of DNA and I can now incorporate it into other pieces of DNA that I've also cut, and you can combine sticky ends and, excuse me, make them stick together. So it's like pretty much little scissors. We have like hundreds of different restriction enzymes, so we can, like, if we've sequenced genes and we know like these palindromes occur before and after, you can expose DNA to different restriction enzymes to cut the DNA in different ways. So this new thing you've probably heard about, CRISPR, what CRISPR does is it's actually super similar to restriction enzymes. So CRISPR is a protein complex, and it also searches for DNA sequences, and then it cuts them at really particular times. The cool thing about CRISPR is that it's more precise than restriction enzymes. So restriction enzymes search for palindromes that are 4 to 8 base pairs long. So 4 to 8 bases long, okay? If I take all of my genome and expose it to EcoR1, it's going to find this palindrome like 20 or more times. So I'm going to cut up more DNA than I intended to if I was just intending to cut out one gene. Versus if you use CRISPR, CRISPR looks for palindromes that are like 25 base pairs long. And if I'm looking for a palindrome that's 25 base pairs long, the chances of that showing up anywhere else besides my intended gene is really low. So CRISPR is more exact in that it'll like just cut out the gene you're looking for versus restriction enzymes, which just like cut kind of everywhere they find these short sequences. So they're really, really similar. So what we can do once we've cut the DNA is we can do electrophoresis. So you can use restriction enzymes, you can cut up the DNA, and then you put that DNA into this gel. So in a gel electrophoresis, you have a power source, and then you have it's an electrical current will be running through a liquid here. So you produce this gel. It's kind of like agar in a petri dish, and the gel will have these little wells. So they're not holes that go all the way through. It'll be like a little divot. And what you do is you take your little DNA that you've exposed to restriction enzymes. So let's say that these three samples, like one is from, let's say we're trying to figure out who the father is, right? So here's like dad one, dad two, child. We take DNA samples from each of them and we expose all of them to the same restriction enzyme. So let's say all of them get exposed to Hindi3. Okay. Hindi3 will cut the DNA at really specific sites. So if I'm the child of one of these people, our DNA should be really similar because I inherited my DNA from them. If our DNA is really similar, then that means Hindi3 should be cutting our DNA in really specific ways. Okay. So you expose the DNA to restriction enzymes. You put those samples in the well. So now in this well is my DNA with the restriction enzyme. Here's dad2, here's dad1. DNA is slightly negative. So if you remember the phosphate group, PO4 minus, makes DNA slightly negative. So when you run a current through the gel, so this is like submersed under a fluid, it's buffer. When you run a current through the gel from negative to positive, the DNA is attracted to the positive end of your gel tray. So the DNA, once you turn the current on, is going to move towards the positive end of your tray. You usually run it for like 5 to 10 minutes. If you leave the power source on too long, all the DNA will actually exit the gel and go into the tray because it's all just going to move to the end until you turn it off. So you usually run it for like 5 to 10 minutes and the DNA will move. So each of these little bands are pieces, fragments of DNA that were cut up by the restriction enzyme. So if I have this restriction enzyme Hindi3, it's going to cut my DNA everywhere it finds that palindromic sequence. And then I end up with all these little bits and pieces of DNA. And what you can look at is you can look at the way things move. So the bands that you see that are further from the well, those are the shortest or smallest DNA fragments because they were all moving for the same amount of time, 5 to 10 minutes, but some of these fragments are moving faster than others. And the reason they're moving faster is because they're smaller fragments. So this would be my shortest or my smallest fragment of DNA. And then the one closest to the well, this is my largest piece of DNA. So it moved the slowest. And then you have all these intermediate ones. Okay. So what we do, so if I'm trying to figure out who the father is for this one, you compare the banding patterns. So this is what we call a DNA fingerprint. This banding pattern, like this would be the child's DNA fingerprint. And you can take its fingerprint and you can compare them. So you can kind of see like these two are similar for these two. They're just like slightly off. So it's possible like this guy is the father. Maybe it's an uncle. So you can figure out relatedness between organisms when you look at the different DNA fingerprints, which is super cool. We can also do this to look at like figure out the genotypes of certain organisms. So you can take their genes, cut them up, and then figure out, are you, oh, so tired, homozygous, dominant, or heterozygous based on how your DNA gets cut, because a dominant gene is going to have a different DNA sequence. So maybe it gets cut, whereas a recessive gene will have a different sequence than a dominant one. So maybe it doesn't get cut because it doesn't have that palindrome. So they can actually sequence and map your DNA fingerprint and determine your genotype for really particular genes. So cool. Electrophoresis is probably the one that comes up the most often, because they want to figure out, like, can you just compare the bands? So you just have to look at the bands. Do they match up? And then what does that tell you about those two organisms? The last one is called PCR. It refers to the polymerase chain reaction. And what PCR does is it just makes copies, as a copy machine of DNA. So a lot of times in lab, when you get samples of DNA, you only get very, very little. So like when we were talking about 23andMe and Ancestry.com, and also just like sequencing the genome of organisms and animals like flies and stuff like that, you're only going to get a really small amount of DNA from your sample. So what they do is they take that DNA that they have, and then they just make copies of it over and over and over. So they'll kind of like find the sequence that they're intending to copy. So like, this is the gene that I want to study and look at. And then there's 3 phases. The first one's called denaturation. So it'll split the strands. Annealing, you add your primers in. So remember, these are RNA primers. So it's like literally just like DNA replication. We're just doing it faster and in a mechanism called a thermocycler. And then extension will extend your DNA. So your DNA polymerase will come in. So you just did replication once, and then you just continue the cycle over and over. So you're just continuously making all these new copies of DNA. And it runs in this machine. Come on, buddy. What is happening? It's right here, called a thermocycler. And what a thermocycler does is it really closely regulates the temperature of the environment of your DNA samples through the 3 steps, because it has to be at really specific temperatures during each step, as you can see right here. So it'll start at 94 to 98 degrees to separate the DNA strands, and then it will decrease to 50 to 65 degrees to add the primers, and then 75 to 80. It's like DNA polymerase, but not DNA polymerase. Taq polymerase will attach DNA to that growing primer. So the thermocycler will just cycle through these different temperatures over and over again, and then you'll end up with many, many, many copies of the DNA. This is like crime scene work too. So if you're on a crime scene and you find like a hair with the root on it, or some skin cells under like a victim's fingernails, then they can use that really small bit of DNA and do PCR. And then once they've done PCR, they'll expose it to restriction enzymes, and then they can do a gel. So they can take like a suspect's DNA from like the scene of the crime and compare it to DNA found on a victim and see if they match. And so that's why a lot of people who were like in prison have like appealed because of the new use of DNA analysis for crime scene investigation. And so like there's some people who were convicted like decades ago before this technique was developed, and now they're being released because using the DNA that was on the scene and on the victim, they found like you weren't the actual person who should have been convicted. So it's kind of cool, the stuff that they're coming up with. So those are like the 3 you really need to know. All these other ones kind of tie into these 3, but these are the 3 they kind of bring up the most. And once again, electrophoresis is the one I've seen the most brought up on FRQs. So just kind of like make sure you know the function of each of these, and you typically use them all together, because typically you're trying to end up here with this DNA fingerprint so that you can actually look at, and then you can also sequence the genome from there. All right, that's it for biotech. Make sure you go watch my video on how to take your AP test because that's coming up for everybody. So I'll go over and watch that, and I'll talk to you guys later. Bye.